Okay, now we've had lots of practice doing Punnett squares and determining outcomes. I had you do so much practice because we need to ingrain that basic way of working with genetics. But now we're going to go deeper. We're going to learn about some genes that don't follow the same pattern, okay? The work that we're going to do, we're still going to do it the same way. Punnett squares are still going to work the same way. The difference is going to be the outcome. And the important thing you need to be able to do is determine what type of genetics are we following to be able to determine the proper outcome. So far, we've done complete dominance or Mendelian genetics. And that is where one gene is dominant and one gene is recessive. It's easy to tell because there's only two possibilities. For example, with the pea plants, you could either be purple or white. The end. That's it. Two options. That's how you know it's complete dominance. But now we're going to learn about a different type of genetic called codominance. Within the grouping of codominance, there are two types of genetics. The confusing thing is that one of the types of codominance is called codominance. So be careful with that, it's a little bit confusing. The other type of codominance is called incomplete dominance. Okay? All right, so let's start with the type of codominance called codominance. So it's not always dominant and recessive. Some genes don't follow that pattern. With codominance, alleles, the different versions of genes, are equally as strong as one another. So one cannot cover up and hide the other. One does not dominate over a recessive. They're both equally strong and they fight just as hard as the other. So what happens when they're equally strong is we have a codominant situation, either codominance or incomplete dominance. With codominance, both alleles are equally strong, and so what happens is you see both at the same time, side by side, in the hybrid or the heterozygote. Okay, so let me give you an example of what this means. Okay, in some species of chicken, you can have an all-black chicken. So they would have two black genes, B, B. You can have an all-white chicken, okay? Two white genes, W, W. Now notice the difference here from what we've done before. One isn't getting a capital letter and the other a lowercase. They're both getting their own different capital letter. And that's because they're both equally as strong. They're both equally as dominant. So they both get to have their own capital letter. So if these two breed, this one's going to give a black gene, this one's going to give a white gene, and so the babies who are heterozygous, one black gene, one white gene, are both black and white at the same time. You see black right next to white in that organism. This is called a checkered chicken. So some of its feathers are black and some of its feathers are white at the same time. This is how you can tell you have codominance, because in the hybrid, you have both at the same time, side by side. So you have a black chicken, you have a white chicken, you have a black and white chicken. So you have the and in it, okay? Black and white. Okay, so this is how you can tell codominance, and this is how we're going to use our letters. They both get their very own letter. You got to be careful. Students love this. This makes sense to them. They love using two different letters, but you've got to be careful because if you're doing complete dominance like we were doing before, you're going to have a big letter and a little letter. You have to be able to recognize the difference in what you need to do. That is so, so, so important. Don't let this take over and then you just hold on to it, because you can't forget the original. That's why we spent so much time on the original. Don't forget it. Here's another example of codominance. You can have blue corn kernels where they have all blue. That's it. They're all blue. So big B. For blue gene. I think they look black, so I used black letters. You can have yellow corn, which is all yellow kernels. So it's big Y. And if you breed them, you get a heterozygote that has both black and yellow kernels side by side at the same time. So this one is black and yellow, or blue and yellow. People call this blue corn. I think it looks black to me, but it's called blue corn. Okay, so codominance, you see both at the same time, side by side. It is black and white, blue and yellow. Okay, so there's an and in there for codominance. Then we have incomplete dominance. Incomplete dominance, both alleles are equally as strong as one another again, but here the difference is in the hybrid or the heterozygote, you have a blending of alleles. So instead of being side by side, they mix together. So for example, black and white, they blend together to make gray. Red and white makes pink. Okay, so we're going to have blending. Blue and yellow make green. So think of paint here. Okay, so you're going to see a blending. They're going to mix together. Okay, they're not side by side. They blend. That's how you know you have incomplete dominance. Okay, when you have a blending in the heterozygote or three very different looking phenotypes. Okay, an example of a plant that does this is carnations. Carnations can be red, they can be white, or they can be pink. So if you think red and white, if they blend together, they make pink. This is incomplete dominance. So if I tell you that you have a red carnation, then it must be big R, because it's incomplete dominance. You can have a white flower, WW. Now, let's do a Punnett square, okay? We're going to do the Punnett square the exact same way as we've done before, straight down, straight across, straight down, straight across, straight down, straight across. The difference here is we're just using two different letters because they're both equally as strong as one another. So I'm going to have my letters fly in the correct place so you know where they're coming from, okay? Straight down, straight across, straight down, straight across, straight down, straight across, straight down, okay? No crissy-crossies. We're doing Punnett squares the same way as we did before. This R goes down into this box. This W goes across into this box. This R goes down into this box. This W goes across into this box, okay, for our babies. Now take a look at these babies. What color are these babies? Hopefully you said they are pink, because they are 100%. But again, look, we did one homozygous type and the other homozygous type, and we got what? 100% heterozygotes. The pattern's the same. The outcome is different because the genes are interacting in a different way this time. So now we're blending to make a pink in the heterozygote. So let's take two of these heterozygous babies and breed them and see what we get. Okay, so heterozygous and a heterozygous. So a pink flower is RW and another pink flower is RW. And again, straight down, straight across. Okay, so this R came down here, this R came across to here. Okay, this R came across to here, this W came down to here. Now what I like to do, because I know a lot of you, you're going to write RW in one box and WR in the other box. That means the same thing. So the way to help you is, before, when we had big letters and little letters, the big letters always went first because they're dominant, always went first. But here, what I like to do is I just like to keep them in alphabetical order. So if you always write your heterozygous in alphabetical order, then all of your heterozygotes will look the same and you won't confuse your brain. Because if you write one is RW and one is WR, you may confuse your brain and your brain will think those are two different things, when in reality they're the exact same. It doesn't matter if you write the W first or the R first. It's the same thing. It's still pink. It's still a heterozygote. Okay, but just to help your brains, since you're new at this, just if you always write them in the same order, it'll help your brain go, oh, those two are the same thing, okay? So just write them in alphabetical order to help you out. All right, now let's look. What color is this baby? RR, hopefully you said red. And then these two babies are the same, they're RW. What color are they? Hopefully you said pink. And then what color is this last baby, WW? That baby's white. Okay, so we have one red baby, two pink babies, and one white baby. Okay, so our phenotype ratio is 1 to 2 to 1. 1 red to 2 pink to 1 white. which is the same as our genotype ratio. 1 homozygous red to 2 heterozygotes to 1 homozygous white. Now, if you think back to our ratios before, these are the same ratios, except for before we had 1 homozygous dominant, 2 heterozygotes, 1 homozygous recessive. Same ratio, except for now the outcomes are different. Okay, red, white, and pink. Okay. Now, with incomplete dominance, we've got to be careful with test questions because remember, I don't get to write the EOC. And there are a lot of different ways that people write incomplete dominance for you. You can do like I do with 2 different letters, and students love that because that makes sense to them. They look different, they should be different. Okay, but again, don't forget complete dominance with the big letter and the little letter. Don't forget. Okay, it represents a different type of genes. But with this incomplete dominance, sometimes you'll see this, 2 different letters. But sometimes you will see where they use 1 letter and a prime. So like R would represent a red gene, and R prime would represent the white gene. So one plant that was R prime would be pink. You don't see that as often anymore. When I went to college, this was the way to do it, and if you didn't do it this way, you were wrong. But now you don't see this one as much. You see this one, the example I just did, quite often. But sometimes you will still see them use a capital letter and a lowercase letter. So big R, little r, and I just noticed I didn't write a little r there. So big R would be red, big R, little r would be pink, and little r would be white. So sometimes you see them use that complete dominance pattern here. So since there's so many different ways of representing it, what you need to be able to do is tell from the phenotypes that it's incomplete. So when they tell you there's pink, there's white, and there's red, you need to be able to say in your head, oh, that's 3 different phenotypes, red, white, and pink. That means this has to be incomplete because red and white mixed together make pink. Okay, if this doesn't make sense to you, if you're totally confused right now, you need to call me over and ask for help, okay? Because you need to be able to tell the difference between complete dominance, where there's only 2 options. You are either purple or white, no blending, purple or white, the end, 2 options. That's complete dominance. Codominance, black and white at the same time, side by side. You are black and white. You have stripes or you have polka dots, same, okay? Same at the same time. Then you have incomplete dominance where you have a blending, 3 different phenotypes, black, white, and gray, okay? Make sure you can tell the difference between the 3 by the given phenotypes, okay? Make sure you understand that. That is super, super important. That is the most important thing about these types of genetics, is being able to tell which one you're using, because they will give you a tricky question where it's supposed to be complete dominance. So they'll be like, oh, they can be black or they can be white. They can't be anything else. They can be black or white. They'll say that in the question. Black or white is complete dominance. But then in the answer choices, they will give you the option of being polka dotted, and they will give you the option of being gray. Both of those are supposed to be obviously wrong to you. They try to trick you like that, so you have to be aware.